On today's show, we will be talking about the Human Genome Project. What is the Human Genome Project? It was basically a collaborative task between nations all around the world to map the entire human genome. A genome is all the genetic material that makes up a human, and this task including mapping the 3 billion base pair sequences in our DNA and the 30,000 known genes. It began in 1990 as a government initiative called the Human Genome Project International Consortium. And now this project was run by someone called Francis Collins. And they ran unmatched until 1998 when a privately funded company called Celera, ran by Craig Venter, came into the research field. Celera had more highly advanced technology than the government, which meant they could complete the project in minimal time. Due to rapid advancements in molecular technologies, electronics technologies, and computer softwares, the project's completion date was accelerated, and in 2001, the first working draft was released to the public. In 2003, the completion of the project was released, and there was a draw that was called between both companies. Now let's talk about the technologies that aided in the Human Genome Project. First, we're going to talk about DNA cloning. First, we need to take the nucleotide sequences of a donor and a vector organism. Then we will apply a restriction enzyme to both DNA sequences, cutting them into fragments. Then we will put both DNA fragments into a test tubes with DNA ligase, which will form the phosphate backbone of the two DNA strands. They will then be connected in the test tube to form recombinant DNA. Now let's talk about Sanger sequencing. First, we'll take our double helix strand and apply heat to denature it. On our template strand, we're going to apply a radioactive primer on the first codon. Then in our 4 test tubes, we're going to apply DNA polymerase, our 4 deoxynucleoside triphosphate groups, and a small amount, 1% of DD-NTP. Then on our nucleotide sequence strand, if we take DD-TTP for example, our DNA polymerase will work across the sequence, adding the DD-TTP whenever it finds a complementary T strand that should be placed. Then from finding these 2 fragments, we're going to use the process of gel electrophoresis. There are significant benefits to the Human Genome Project, one being in medicine. Through the formation of linkage maps, we can now identify which genes cause certain diseases. This helps doctors prevent, diagnose, and treat common diseases. It also helps us in the process of inheritance. Say my father had the disease cystic fibrosis. I can now, through advancements in the Human Genome Project, work out the percentage that I would get this common disease. Then I can get certain treatments, such as gene therapy, which is basically removing the faulty gene from my DNA sequence and replacing it with a normal or healthy gene. Human Genome Project has also benefited in our biological understanding in evolutionary similarities. By comparing a little human genome to that of other organisms, we can work out certain similarities. If we take the mammals for example, we can work out that we have 14,000 out of our total 30,000 genes in common, which may suggest a common ancestor. Also, if we look at the example of chimps, advancements in the Human Genome Project has allowed us to understand that 98% of our total genome is exactly the same as that of chimps. There are also limitations to the data collected from the Human Genome Project. The data collected doesn't actually give us the specific function of these genes. Thus, scientists must still do the manual matching of these base sequences to the specific known gene. There is still a lot of manual work that must be completed by science after the Human Genome Project. The data also doesn't take into account environmental factors which can impact upon the gene that is being actively expressed. For example, the nutritional lifestyle of an individual can impact upon, say, the tau gene being expressed. Thus, scientists can't actually identify the phenotype and genotype of an individual after they've been already born. Data obtained also doesn't show the polypeptide chain which will be synthesized or what protein will be created. This means that scientists have little knowledge about where in the body that these genes will be actively expressed. This means that scientists must still do a lot more research into the total understanding of our human genome. Now let's talk about genetic linkage maps. Genetic maps are the frequency in which recombinance occurs during the process of meiosis in cell division. Now, what recombinance is the rearrangement of heterozygous homologous chromosomes into new combinations during meiosis, which shows us the relative distance between genes on any given chromosome. In 1913, Mendel's student Sturtevant theorized a formula to work out how genetic linkage maps can identify the position of genes on a chromosome. He used this formula: the number of recombinant offspring over the total number of offspring times 100. If we take this set of data, for instance, and we look at the genotypes of the offspring produced, we can see that it comes in two forms: either the homozygous form of the linked genes or the heterozygous or recombinant form of the linked gene. If we then substitute this data into our formula, we work out there is 11% recombinance in this generation. If we then look at our other formula, it says 1 map unit equals 1% of recombinance. So then we can deduce that there is 11 map units between these two linked genes. What Sturtevant deduced from his research was that the greater the percentage of recombinance within a generation, the greater the distance between genes on a chromosome. The theory behind this was that the greater the distance, the more chance that chromatids would cross over in this region between the genes. So if we take these two, for example, with the greater distance and the less distance, this one has more chance of recombinance occurring, whilst this one only has a small chance. Linkage maps, however, aren't a viable tool in the complete mapping of the human genome. The reason is that genetic linkage maps don't give an exact location of where the genes are on the chromosomes. They don't tell us whether the gene is present within the long chromatid or the short chromatid. This is of significant hindrance to the project, as when scientists are analysing certain diseases or traits, scientists must still do the manual base sequencing. Another reason why we can't use linkage maps is because in linkage maps they don't include these introns. And if we look back to the goal of the Human Genome Project, it was to sequence the 3 billion base pairs. And using linkage maps, it doesn't include these sections in between these genes, which is crucial to the total understanding of our human genome. Genetic maps don't give us an indication of the exact base sequences of all genes, which leads to my argument that physical mapping is more appropriate for the Human Genome Project. The Sanger method, which we looked at previously, allows us to identify the exact base sequences of genes which we are studying. Ultimately, the project has revolutionized our understanding of biological thinking into the world of biology. Thank you for listening to me, and until next time, adios.